Let's talk briefly about the technology in a very simplistic terms. I think simple is elegant, and simple is beautiful. And then I talk about some of the applications. How many of the applications of CRISPR-Cas is indistinguishable from a conventional breeding product, right? And at the end, I'll give you a couple of examples. I'm going to today focus mostly on corn, but we are doing the same thing in rice and multiple other crops. So what we want to do, we want to provide the farmers with best quality seed, so that they are able to do climate smart agriculture, right? The farmers face every year, the weather is changing, the climate is changing. So we want to have seeds which can perform in these adverse conditions, right? It's not very easy, right? And CRISPR-Cas allows you to do that with this advancement in the breeding technology. As Dr. Rao said, there is a lot of excitement, and you can see the number of papers. In 2015, it was about 1200. Like one of my colleagues sent me this latest information. In 2016, there are 2100 papers on CRISPR. This is what, and if you compare this with RNAi, I think the maximum that we could reach was 700 in a year, right? So this technology provides tremendous excitement. Not only in the scientific, the best thing is not in the scientific community, but also in public in general, right? That is one of the most exciting part of the technology. So, not surprisingly, this has caught imagination beyond the lab, right? There is an article in Economist editing humanity, and one of the questions that came was regarding that, This CRISPR-Cas craze is not without reason. Right? It's not that genome editing technology is evolving now, right? It has been in the, it is there for quite some time. But what CRISPR-Cas provides is a tremendous opportunity, and the beauty is in the simplicity. Now let's look into what are the options available. So we didn't have a lot of work around zinc finger nuclease. We don't have access to work on that one, so I don't have as much information. But this is in-house research that we did in all these nucleases, right? So whether it is zinc finger, meganuclease, taline, as well as CRISPR-Cas. And as you can see here, from the time of efficiency, quality, cost, as well as technical flexibility, CRISPR-Cas is the best so far. Now, why is it so? So as Professor Venkateswara very elegantly demonstrated, the first three of them are nothing but engineering protein. And all of us as students know engineering protein is much more difficult than engineering RNA or DNA, right? If I can use the analogy of cell phone, so all the earlier generation of genome editing technology, whether it's zinc finger, meganuclease, taline, gave you opportunity, but it was like smartphone. So for example, if you want to call somebody, a person X, you need to have one smartphone. You call another person, you have another smartphone, right? Whereas with CRISPR-Cas, it's like a smartphone with a directory, right? So what you need is a guide. Just design the guide. Cas9 is the same. So with the same cell phone, smart cell phone, you can call multiple people, right? Which was not possible earlier because you have to engineer protein, and we know engineering protein is much more difficult than engineering RNA or DNA. I think that's the best advancement that is provided with CRISPR-Cas. The process happens in nature. So what is happening when the plants or animals or human beings, we are exposed to different conditions, whether it is internal stress by reactive oxygen species and other stresses, or external stress, DNA break happens in nature, right? And it gets repaired. Plant has two kinds of mechanism. One is non-homologous end joining, which is less idle, so it creates mutations, or homologous end joining, both of them, right? As Professor Venkateswara again very elegantly mentioned that the frequency of these two processes are different, but they are of immense use. So this is nothing different than what is happening in the nature. So what CRISPR-Cas does, in CRISPR-Cas system, you have a guide RNA which targets the Cas9 protein to the target sequence. So in essence, what you need is a guide RNA which is targeting to a particular region in the genome, and the Cas9 is there, right? So this guide RNA takes the Cas9, creates a double-stranded break, and then depending on what cell does, not naturally, the double-stranded break is happening. it can get repaired either, as I said, by non-homologous end joining, in the process creating insertions, deletions, or if you can have a homologous end joining where you may give a template where you can put different gene or DNA into it, right? So depending on what kind of phenomena is happening, you can have different applications of CRISPR-Cas. For example, you can have a delete, which is basically non-homologous end joining. There is a double-stranded break, and cell repairs it. In the process of repair, it may incorporate a DNA or a delete a base of DNA, thereby creating mutation. Right? So you can have a single base pair deletion by changing a protein, by changing the nucleotide, or you can have a edit. So in this case, you can provide an external template which will allow you to change the nucleotide sequence, or for example for the protein sequence. For example, if you know that this particular enzyme, the active site is this particular base, so you could change that and change the activity of that protein by changing a single base pair mutation, right? So that is also possible. The third example is search and replace. So basically, this is in this kind of situation, you take a DNA sequence from another place in the genome and put it in a different context, right? And I'll give example of the first kind and the last kind. So depending on all these phenomena, you can have these kind of outputs where the plant may have susceptibility, or it becomes resistance and tolerant to drought and disease, and so on and so forth. Now here I will take a pause and try to introduce you what breeders have done over the years, right? So breeders have looked for variation in the genome, in the plant species, and they have selected it, and thereby creating new products. Now this was very, very surprising for me, and I don't know how many of you know, at least I didn't know. All these species that you see, whether it is kohlrabi or kale or broccoli or Brussels sprout, cabbage or cauliflower, these belong to the same species. So breeders have selected, depending on the modification, either it is in stem or leaf, lateral bud, this is the same species. So for many of the applications of CRISPR-Cas, the final product is indistinguishable from a product that can be generated by CRISPR-Cas, right? So if you are creating a single base pair mutation, that naturally happens. So when the plants are grown, they get exposed to changes, even naturally, right? There is a changes, like the deletions happen, SNPs are there, copy number variations are there. There is a huge number of changes that is happening in the genome without doing any intervention. Now let's look into traditional breeding. So one has to understand that the germplasm is key, right? So germplasm can be global, but it has to be adapted to the local condition. You can have a great germplasm, it can have very good phenotype, from one condition, but if it doesn't give you the same phenotype in a different condition, it's a useless thing, right? So we need to adapt the germplasm to the local condition. So you may have a plant which is drought tolerant, but the other characteristics are not good. So it is not adapted to a particular region. For example, it's a US germplasm you want to grow in India, it may not do well, right? So in case of, and then what you need to do in the traditional breeding, you need to cross it with the Indian germplasm, right? And then keep on doing the back process. At least for in this case, the example I have taken is 5th generation, and you can do simple math. So even after 5th generation, you still have 3% of the donor genome. Now, as a science student, we know even 3% of a few MB genome is a big number. You can still have yield drag. So it's not good enough, right? So what CRISPR-Cas allows you is to do the desired changes in the elite germplasm itself, because you are going to do small mutations in an elite background. You do not need to have the backcrossing that is required in traditional breeding, right? So you go and do the modification in the final genome. So you have a plant which is of lower quality but tolerant to drought. You edit it, make it finally the higher quality plant with drought tolerance. This is what the CRISPR-Cas technology allows you to do. Now, what are the critical success factors? It's not that only the CRISPR-Cas technology is important. There are other factors important. You need to really understand your genetics, your germplasm. Without that, you will fail. Unless you know the exact precise sequence of a gene, you can't edit it, right? So what is there probably is a model sequence, right? For example, if I take example for rice, the sequencing that has been done for Nippon barley, right? So if you design your targets based on that sequence, you may not be able to then edit because you don't know what germplasm you have, right? So it is very important it is very important that we get the right germplasm and genetics. So sequencing information is important. Equally important is the ability to deliver molecules very precisely, right? So you have to have a really great system to transform. It may be, you may be able to transform model system, but not the elite lines, right? So that is another area. The third one is advances in CRISPR-Cas technology. And what we have been working on all these three, and we have great breakthrough in all these areas. So you may have seen there is a recent plant cell paper that came last year. We have the ability to transform almost any germplasm, right? And that has happened because we have been working on this for a long time. And also we have been working to have superior activity and targeting specificity for CRISPR-Cas. And we are collaborating. We are doing a lot of in-house work. We are collaborating with many different institutions and companies for that as well. So what kind of product, I have 5-minute time, so I'll try to wrap up quickly. So what kind of product you can have a number of different products which is listed in this slide. I'll move on to the next one. So this is the example, I'm going to cite two quick examples. One is Vaxicon. So what is there in the Vaxicon? Please don't get into the technicalities. The yellow dent is nothing but the commercial corn, and the Vaxicon is a variant of a corn which doesn't have amylose. Okay, it only has amylopectin. So it gives a different kind of this candle wax like appearance. What is the utility of this? We really understand this because our company is working on genetics for 90 years. Globally in India we are over 40 years old. Okay, so we have a really strong product familiarity. Pioneer is one of the leading supplier of Vaxicon. And this is an identity preserved product. Like maybe in the discussion we will touch base on what it is. Let me quickly go to the example. So this particular product has got the utility in food industry, paper making, corrugating, textile as well as adhesive, right? So what we have currently Pioneer is the leading provider of Vaxicon in the world. So what has happened in this particular case? This is a mutation that originated more than 100 years back, and there is problem with this particular product. So it can never we can never do this in the elite line. So there is a 2 to 3 year gap, and there is a yield drag. Whereas the new generation Vaxicon that we are trying to do is a full length deletion, in comparison to the current one, and we can do this in the elite variety, right? Now this is a recessive mutation, so we have to do for both the parents, and that's what we are working on. What is the regulatory status for this particular product? As you know that I'm pretty sure Dr. Roy is going to touch base on this one. The regulations are evolving in different countries, so there is a lot of discussion happening. We have submitted our application to USDA, and we have got confirmation from USDA that this particular vaccine product is not regulated under the USDA regulatory services. 1 minute. So I gave you an example where I talked about the SDN1, the deletion. Now in this example, I'm going to give you an example of SDN3, which is basically what we did. We have done a lot of work in this particular gene. We know that if you in a transgenic context, if you mildly overexpress ergoside gene with a maize promoter, different maize promoter, you can get tau problem. So in this particular example, what we have done, we have created two different versions of ergos gene. In both the cases, in one case we have inserted a promoter which is a maize promoter next to the ergos gene. In another one, we have deleted the native promoter and put that another maize promoter. So which is version 1 and version 2. In the normal course of action, so you can see the expression of this particular gene is very low, whereas in both the genome-edited lines, the green and blue, you get higher expression. And when you do the drought tolerance phenotype, look in the field, under flowering state, you get 5 more bushel of yield, and 1 bushel roughly is about 25 kilo, right? And you can see not only in the drought tolerance, under optimal condition also it gives you more. So I hope I am able to convince you that this technology has great potential, and we are working very rapidly on all the crops. This gives us enormous opportunity. So again, I would like to take this opportunity to thank the organizers, Dr. Rao, for giving me the opportunity. I also wanted to thank my Pioneer colleagues who have been putting a lot of effort in all the crops, and also for all of you for showing this excitement for this new technology. This is going to be a game changer, and the country can make a lot of progress with this. Thank you so much.